Alright, this is the video for perhaps the most important chapter in your book, Genes to Proteins, which lays a very solid foundation for a molecular understanding of genetics and evolution, which you need to have. I want you to take extra time this week to ask me questions, come to my office hours, and make absolutely certain that you understand this material because we'll be building upon it for the rest of the semester. Okay? Now, in Chapter 8, your book presents these driving questions. In this video lecture, I'm going to describe mostly the way a protein shape is determined, how genes are expressed, which is extremely important, and we'll talk also about GMOs, which are genetically modified organisms. I'll spend some time talking about these two things, not as much as your book, but we will talk a little bit about them. I do want you to have a more conceptual overview of genetically modified organisms to prepare you for 1, your discussion, 2, your Unit 2 assessment. You're going to be looking at GMOs again, and even just the next time you're in the grocery store, I want you to understand what you're looking at. So to talk about the way genes produce proteins and form proteins, we need to understand what exactly a gene is. We've got to be able to define it. So a gene can be defined several different ways, but for the purposes of this class, we'll just define a gene as a nucleotide sequence which encodes a complete protein, or a trait, sometimes this can be called. A trait would be something like how many fingers you have, or the color of petals that a flower has. Those are all determined by proteins. As we learned in Chapter 7 in this last video, sometimes an error can occur during DNA replication, and that results in a mutation. If this mutation doesn't immediately kill the cell, sometimes it does result in cell death, but sometimes it doesn't, and it can produce a slightly different nucleotide sequence or a slightly different version of that gene. And that's called an allele. So for example, here we have a flower example. We see 2 chromosomes over here. The same place, the same loci, it's called, or locus, loci would be plural, same locus on each chromosome, these homologous chromosomes that are the same pair, maybe they're chromosome 1, that same locus is where the gene is, the gene that codes for the same trait, the color of the flowers. But these are different alleles because this one is a little bit different and it encodes for purple flowers, while this one down here codes for white flowers. They are the same gene, but different alleles. The way people use the word gene in everyday conversation, usually you'll say like, oh, you have the gene for blue eyes, or you have the gene for freckles. That's not actually true. You have the gene for hair color or skin pigmentation, which in humans, these 2 traits are bad examples because there are multiple genes that inform those traits. But what you really have are different alleles, the same gene. Everyone has some gene for hair color, but everyone has their own unique alleles to give them their own unique hair color, for example. This we'll talk about a lot more in Chapter 11, but this was a natural way to introduce the idea, the difference between a gene and an allele. They don't mean the same thing. Okay, a gene is expressed through 2 steps, transcription and translation. Transcription occurs where the DNA is. So if you're talking about a eukaryote, which we will for the rest of this unit probably, that's the nucleus, where the DNA is, where the chromosomes are. The product of transcription, which is called messenger RNA, travels outside of the nucleus to find a ribosome, which can be in a very number of places, and that's where translation takes place. The product of translation is a protein. So this is how genes inform the production of proteins. We're going to go through each of these steps in more detail and talk about some of the molecules that are involved in what exactly it is that they do. Transcription, or to transcribe, means to make a copy. It's a little bit different from replication, though. So in biology, transcription means that a copy of the genetic information is made, and this copy is a molecule called messenger RNA, or just mRNA. To transcribe mRNA, the DNA double helix is unzipped, just like during DNA replication. And recall that during DNA replication, DNA polymerase makes a copy of a template strand, which we have down here, by matching complementary nucleotides to that template. The difference here is that the new molecule being made is RNA, not DNA, so that enzyme is called RNA polymerase. So, like I said, RNA polymerase follows the same base pairing rules, the same complementary rules, but with RNA, instead of matching thymine to adenine, a nucleotide called uracil is bound to adenine. So in a molecule of messenger RNA, or any RNA, you don't have any T's, you have U's instead. The other pair is exactly the same, guanine to cytosine, and so forth. So once this goes along and is transcribed, it is kind of unzipped another way here so that you've got messenger RNA as a single strand, and eventually the DNA will wrap back up. I want to take a moment to talk a little bit more about this, because RNA polymerase has a specific site on each gene that it binds to. This picture isn't very good for showing it, so I'm going to do my best. Bear with me. So on this template strand, if you can imagine following it along, I'm just going to unzip it a little bit more here. So this is the template strand. There was at some point something called a regulatory sequence. These are base pairs, by the way, just the one side. And so some of that DNA sequence, those base sequences, will code for that regulatory sequence, which just is an area on right in front of a gene where the RNA polymerase kind of clasps onto the template strand and starts copying from there. So I'm going to label that with an R because it's regulatory sequence. The rest of this stuff, we'll move to this color. The stuff that actually gets transcribed into messenger RNA, which would have gone up this way a bit, this is called the coding sequence. So I want to label that with a C, coding sequence. And at some point, this RNA polymerase will run into a shorter sequence that tells it to stop. That's a stop codon. It wouldn't be this short. I'm just trying to make it simple. So we'll just label that with an S. There will be a stop codon. Sometimes it's a little bit longer than just the 3 pairs that I'm about to tell you about. But the point is there is a place right after the gene that tells RNA polymerase to stop copying. And there's a place in front that tells it to start copying. That matters when we talk about GMOs. So I wanted to tell you about it. Okay, so once that messenger RNA is transcribed, and it being only the length of one gene, it's able to leave the nucleus and go find a ribosome. So messenger RNA is still involved in translation, and this new molecule called a ribosome, it's a structure made up of a couple different molecules. It's got these 2 subunits, a larger and a smaller subunit. Anyways, what a messenger RNA needs to do is find a ribosome. These can be floating around in the cytoplasm. They can also be on the rough endoplasmic reticulum, the rough ER. And depending on the cell and the type of protein that needs to be translated, certain locations are better. Once the messenger mRNA reaches a ribosome, the ribosome takes the messenger RNA between these 2 subunits, sort of like a fist holding a rope. And there are 3 sites inside the ribosome where the nucleotide sequence on the messenger RNA is read. The sequence is read 3 bases at a time, like it shows here. There's 3 bases, the next 3 are read together, and the next 3 are read together. These 3 bases together are called a codon, and they are the complement to another group of 3 nucleotides on another molecule called transfer RNA or tRNA, and this group of 3 nucleotides on the tRNA are called an anticodon. And again, they're complements to the codon on the messenger RNA. So tRNA, transfer RNA, is floating around in the cytoplasm, and it carries or transfers amino acids. Now remember that there are 20 different amino acids. This means that there are about 20 different tRNA molecules, and each one of them has a unique anticodon and carries a unique amino acid. Therefore, amino acids are brought to the ribosome in an order according to the codon on the messenger RNA. Within these 3 spots on the ribosome, the transfer RNAs deposit their amino acids. So they come in sort of in this order. This is in the first location that this tRNA will be in. It matches to the codon on the messenger RNA, and then it moves into this position. And there are 3 of them doing this at a time. And there, a chemical bond is formed linking the amino acid from one tRNA onto the other tRNA. And here's the 3rd position. After this 3rd position, the tRNA leaves without its amino acid. It leaves its amino acid onto this growing chain, this new protein, and that tRNA can go get a new amino acid and bring it back if needed. This is how chains of amino acids are made. Meanwhile, there's more tRNA coming in, so you can get some pretty long chains going on. So this growing chain of amino acids, get rid of those drawings, this constitutes something called the primary structure of a protein. So in the other image, it's just showing, spheres, just little circles for each amino acid. This is an actual chemical diagram, but here we have a chain of amino acids, just a little bit more complicated of a drawing. That's the primary structure. So these amino acids have different atoms on them. They're polar, they have different charges, and they interact with other amino acids at different locations on that chain, and they can form different shapes into these secondary structures. There are 2 basic different secondary structures that can form. One's called an alpha helix, and it's just a single-stranded helix, similar to the way DNA forms a double helix, but this is only 1 strand. And there are also these things called beta pleated sheets, and it's kind of this zigzaggy formation that folds up on itself. So you've got these different width. It can grow this way even more, of just sheets of secondary folding within this protein. That's the secondary structure. This diagram is a little bit misleading in that it devotes a lot of time to secondary structure, and then when you get down here to tertiary structure, it looks a lot smaller, but in reality it's a lot bigger. The tertiary structure is when you've got these secondary structures, either these spirals or these sheets forming, they fold up even more into this pretty complex folding of a protein. Okay, that's called a beta polypeptide sometimes, but don't worry about that. Now, sometimes this can even get even more complicated. You can have different proteins, completely different chains folded up into their tertiary structures that come together to form a highly complex, very large molecule, a quaternary structure protein. An example of this is hemoglobin. That's what they're showing here. These heme groups are where the iron is. That's the function of hemoglobin, if you've forgotten, is to carry oxygen, gaseous oxygen from your lungs through your blood to all of your cells that need oxygen for aerobic respiration. Now, it's very, very important to remember that this shape, which is extremely complex, is absolutely imperative to the function of a protein or an enzyme. If the shape is wrong, it might not be able to do its job. And it's important because that shape is completely dependent on all of these previous structures. The quaternary structure will not be right if tertiary structure is wrong, and tertiary structure will be wrong if the secondary structure is wrong. The secondary structure will be wrong if the primary structure is wrong, and that is all dependent on the messenger RNA bringing an unmutated sequence of information with these nucleotides from the DNA. So if there's a mutation in the DNA, it gets carried through the messenger RNA to the growing amino acid chain, and it messes up the entire protein. It can, at least. Now that can have some pretty big problems. In this hemoglobin example, if hemoglobin is shaped wrong and it doesn't carry oxygen, hemoglobin is so large that it actually will even change the shape of the blood cell that it's in, and it can form something that you've probably heard of called sickle cell anemia. Instead of having these nice round red blood cells, you have these sickle-shaped red blood cells that don't carry oxygen. And so this can be very painful and without treatment and can result in death even. So it's a problem. That's just because there's a mutation in the DNA, which leads through this cascade effect to a very big change in a very important molecule. That being said, sometimes a mutation can even be good. Maybe a person might have a mutation that leads them to having these beautiful long eyelashes, and if you're a nomad, whatever, 20,000 years ago, that helps you keep debris out of your eye through the blowing sands. It also makes you pretty attractive. That's something called sexual selection. It is a thing. But sometimes the mutation doesn't really do anything. It's not harmful, like sickle cell. It's not beneficial, like eyelashes, for example. Maybe a person might have a mutation that leads to them having red hair, and then whatever, who cares? It doesn't really confer much of a benefit. Or maybe blue eyes, or whatever. In any case, the way that an organism looks and behaves depends on their protein expression, which depends on their DNA. And the relationship between DNA and proteins and the way an organism looks and behaves, that's called a phenotype. That is kind of the subject of the rest of the unit, so it is very important. Now we can apply this. We have learned as a species to kind of mess with DNA for a while. For thousands of years, up to 10,000 years, we've been domesticating organisms. We've selected for the largest ears of corn in our food, the fastest horses, or even the cutest dogs from wolves. But more recently, we've gotten really good at this. We've discovered ways to take the genes from one species, called a donor species, and put it into a brand new species, an unrelated species, called a transgenic species, right? This actually does happen in nature. It's called horizontal gene transfer. I mentioned it once when we were talking about the photosynthetic sea slug. It's a natural example of horizontal gene transfer. Bacteria do this all the time. They're really good at it. So it sometimes happens with humans as well. A retrovirus such as HIV, it puts its own genome into the host genome. That's why it's called a retrovirus. I could talk all day about that. Never mind. Occasionally, there's a messed up HIV that's actually carrying DNA from whatever host cell it came before. If it just kind of packaged the wrong DNA and then deposits that can happen too. So horizontal gene transfer does happen in nature, and it's not always a problem. But what we've come to realize is that we can do this on purpose and very directed. And so this is called transgenics, moving genes across species. This creates a transgenic organism, or also called a genetically modified organism. Those are synonymous. They mean the same thing. So a common GMO that I don't know if you've heard of this, these are really cool. There are freeze-tolerant tomatoes that produce just a natural antifreeze, so you can plant them a little bit earlier, and if you experience a freeze, less of your crop is completely destroyed. And this gene that produces this natural antifreeze, which is a protein, it just lowers the freezing temperature of whatever fluid it's in. That gene comes from a fish that lives in Antarctica. And they live in sub-freezing waters, and so to keep their blood from freezing and their cytoplasm from freezing, they create this molecule, this natural antifreeze. So they can take that gene from that fish, put it into a tomato, and when that tomato plant grows, it's a little bit more resistant to freezing. So this can result in much higher crop yields and feed more people. GMOs have a lot of uses, so that's one. Another example is golden rice. It's one of the most famous ones. This is rice that's modified to produce more nutrients, such as vitamin A. And that is a huge benefit to people, especially people living in third world countries, living in poverty, where good nutrition, or even just enough food at all, it can be very difficult to come by. So we can feed the world more nutritious food this way. There's also the first transgenic organism was E. coli, which is a bacteria. In 1978, E. coli was first modified to produce insulin. They took the human gene for insulin from the healthy human, put it into E. coli, and that E. coli started making insulin, which is another protein. And then in 1982, the FDA approved the use of genetically modified E. coli for production of insulin for therapeutic uses, for medicine to give to diabetic patients. Your book tells a very similar story with the goats, which are modified to produce the antithrombin protein. So there are, of course, therapeutic uses to genetically modified organisms as well. And there's another more controversial example, the Roundup Ready maize or Roundup Ready corn. These crops are modified to resist exposure to Roundup, which is an herbicide. It kills weeds, but corn is also a plant. It's pretty related. So if you just spray a bunch of Roundup on a field of regular corn, it'll kill your corn too, and you don't want that to happen. So in order to be able to spray fields to kill weeds, they produced Roundup Ready corn, so you can spray the field, get rid of the weeds, but your corn is fine. This one has some pros and cons. There was a study done, and if you Google like GMOs, it's like the first or second hit, it's right away. There was a study done in Europe on rats that were fed Roundup Ready corn, and they claimed to find an increased risk of tumors in these rats. I'm not going to go into that, but you should probably look it up. And if you go find that study, make sure you find the original peer-reviewed published article. And kind of a spoiler alert, if you go find that original PDF, you'll find that much like the Wakefield vaccine study that we looked at in the first week, it has also been retracted because of some of the things that these scientists did and how they were making claims based on that evidence. I'll let you look for yourself, and there's a lot of explanation for why. Now, I just wanted to bring that up so that we can talk about the safety of eating genetically modified crops. So what happens when you eat GM food? Let's talk about this, right? So Roundup itself is a toxin. You don't want to eat herbicides. Eating unwashed corn that's been sprayed with Roundup, that'll probably make you sick. As a side note, you should always wash your fruits and vegetables anyways, whether they're organic or not. The ones that are non-organic can have pesticides on them in low levels. Just wash those. And even if they are organic, they are completely covered with bacteria, I guarantee it. It's just rinse those bad boys off. But anyways, if you get sick from eating corn or something that's covered in herbicide or pesticide, that'll make you sick. But is that because it's genetically modified? Oh, no. It's because you ate a very toxic chemical that's designed for killing life forms. Now, the DNA that makes up the gene in the corn that makes it not killed by Roundup, that's just DNA. You're just eating all of the normal macromolecules and macronutrients in that corn if you wash it off or whatever. Or if it wasn't sprayed, like in the study, they didn't even spray it, they just, anyways. So that DNA is catabolized when you go to eat it. You digest that DNA, and then you've got just monomers, nucleic acid, nucleotides just floating around, and you can use them to make your own genes. But you won't suddenly like have that trait. You won't be resistant to herbicides, or you won't become freeze tolerant like the tomatoes. It's just DNA. That's all you're eating. Just the way that you don't get any traits from any other food that you get. If you eat a bunch of bananas, you don't become a banana. This is basic, simple stuff that you already intuitively understand. A GM crop is exactly as safe as a non-GM crop, all other things being equal, like contamination from bacteria, pesticides, herbicides, and so forth. So when you do your discussion this week, and in a couple weeks when you do your Unit 2 assessment, I want you to think about that and make sure that you critically evaluate any evidence you encounter, because there's a lot of BS out there. Make sure you critically evaluate everything anyone tells you, including the spiel that I just gave you. Understand the processes that are going on here so that you can write about it. Okay, as I said, the material from this chapter is so important. That's why this video is probably longer than usual. But we can sum up the main points this way. You should know how the shape of a protein molecule is determined, the steps included in gene expression, including where those things happen, some in the nucleus, some at ribosomes, in the cytoplasm somewhere. And you should also know a little bit about transgenic organisms, or GMOs, including what they are and a couple of uses for them. After watching this video, you should take the Lecture 5 quiz. Check out, there's an online GMO activity as your assignment for this week, and make sure you contribute to the discussion. There, like I said, it's most fun if there are some, impassioned responses in these discussions, but remember to keep it civil, and even more importantly, evaluate your sources for credibility and think critically about the evidence that you're presenting or that someone else is presenting. This is a very good chance for you guys to learn to argue scientifically. That doesn't mean fighting, it means using evidence to support your own claim. And make sure that you do good research. Like I said, there's a lot of BS out there on this topic of GMOs. So make sure you're looking at good sources that are credible. You might need to review week 1 stuff if you're a little shaky on what credibility is and what strong evidence is. And just hang on to those sources, because your Unit 2 assessment is going to expand on this and you might need them again. Okay, have a great week. I hope you learn a lot.